Hello and welcome to Health Professional Radio. I'm your host, Neil Howard. Thank you so much for joining us for another segment. In this segment, we're going to be speaking with Dr. Harlan Robbins. He's joining us here from Adaptive Biotechnologies to discuss why vaccine sponsors need to rethink vaccine design to take advantage of the T-cell immune response to COVID-19 variants. Welcome to Health Professional Radio, Dr. Robbins. Thank you so much for taking the time.
Thank you so much for having me.
Well, give us a bit of your professional background and then provide us a brief summary of Adaptive Biotechnologies and your role at the company.
Sure. So I was an academic for quite a while. I did my PhD in a theoretical branch of physics at the University of California, Berkeley. And then I went on to do a postdoc at the Institute for Advanced Study and switched to computational biology. Then after that, I took a faculty job at the Fred Hutchinson Cancer Institute in Seattle, where I then became the head of computational biology for that institution. And then my brother and I spun out a company called Adaptive Biotechnologies from the research that I was doing with some coworkers at Fred Hutchinson Cancer Institute. And so that leaped into Adaptive, and that we found in late 2009, and as of now, I'm the Chief Scientific Officer at Adaptive.
Well, Dr. Robbins, tell us a bit about your T-cell compounds.
We took all of our technology and wrapped it up into a product that's available commercially to assess the T-cell response, and in particular for whether or not you have had past SARS-CoV-2 infection and based on and or the vaccine based on your T-cell response, and we call that Tdetect.
Let us in on what COVID-19 vaccines were originally designed to do and why.
So the primary direction that all vaccines that we've had successfully on the market, including COVID, have been to induce what's called a neutralizing antibody response to prevent the virus from initiating an infection. And so basically for SARS-CoV-2, the easiest way to do that is to basically block how SARS-CoV-2 gets into lung cells. How does this virus get into lung cells? And so the tip of the spike protein on the surface of these viral particles binds to a receptor on lung cells called the ACE2 receptor. And it's that binding that allows the virus to infect lung cells. And antibodies, which are excreted from B cells, can stick onto that spike protein and prevent the virus from getting into the lung cells. And that's how this vaccine was designed to work. So that's why all the vaccines are made up primarily of the spike protein. Mm-hmm. And they try to mock what the actual virus looks like, and then your antibodies naturally are formed to bind to that position in the vaccine portion, which then looks enough like the actual real virus that it then blocks the real virus as well.
Now, I said in the outset that we were going to talk about taking advantage of the T-cell immune response to COVID-19 variants. Why has the immunity focus of COVID-19 vaccines shifted now to T-cells? So
as I was saying, with these antibodies, they bind to that tip of that spike protein and then attempt to neutralize the virus. Well, the vaccine spike protein and the actual virus are no longer the same because the virus keeps evolving. So the vaccines are still doing what they're supposed to do. They induce a really good set of antibodies that bind to the spike protein from the original strain, in this case, the strain that we had from Wuhan, a couple of years ago. But that's not what the virus is anymore. So the antibodies that are being induced no longer bind to and neutralize present circulating strains of the virus. So Omicron, we're losing over 99% of efficacy of neutralization from these antibodies. So why are vaccines still having some efficacy? The reason is that you have multiple parts of your immune system. Your immune system has, even your adaptive immune system has and I'll just give you the broad dogma that antibodies prevent you from getting infected, as I just described, but that T cells clear infection. They're the ones that prevent you from getting really, really sick. Mm-hmm. So at this point, we're not doing a very good job anymore of preventing infection, as I'm sure you all know many people who were vaccinated that then ended up getting Omicron. But it's still doing quite a good job of preventing people from getting severely ill and dying. And one of the ways you can see that, I mean, look at what's happening in Hong Kong, where the vaccination rate was lower and across the board, and they prevented the earlier strains from occurring, so people didn't have much immunity. Omicron's still being quite virulent in there, whereas here it hasn't been, because people either have high levels of immunity in one way or the other. So the T cells are what's preventing people from getting ill, and because T cells aren't just binding to and causing effect on that one little tip of the spike protein, they can have impact across the entire genome. The variations that we get don't really escape the T cell response, meaning that the new strains are still, even if you induced an immune response from an old vaccine from the Wuhan strain, it should still have quite good efficacy on the T cell side against Omicron.
Do regulatory agencies look for T cells in addition to antibodies in these vaccines? Do they require an assessment for both?
So not really. Of course, you can submit whatever data you want, but it's not used as a standard for acceptance of a vaccine. So, for example, you could have almost no or minimal T cell response from your vaccine as long as you had a good neutralizing antibody response. That vaccine would be considered acceptable. And if you had no neutralizing antibody response and really good T cell response, that would not be considered acceptable. It's not accepted as a standard for decision making by the FDA at this point, which really needs to change.
Is there a reluctance based on the fact that they're T cells? We hear a lot about the benefits of T cells, but we also hear that they're very costly. Is that the case?
So it used to be, and the issue is that antibodies are excreted into your system by B cells. So they're just proteins floating around that you can just be measured in the blood, and it's really cheap and easy to find them and measure them and measure properties of them. The T cells, unfortunately, so T cells are only act locally, meaning that if you want to know what a T cell is doing, you have to look at the T cell itself, not at something excreted from them. And the traditional methods of measuring what T cells are doing are based on functional assays on live cells. And that is extraordinarily expensive and very hard to standardize. However, that was what we set out to change, which is, could we look at a property from these T cells that didn't require live cells, that was ultra stable and much less expensive? And we were able to do that by looking at specifically the DNA. DNA is really, really stable. We look at the DNA of these T cells and are able to assess the property about what they're binding to. In other words, we're able to pick out the T cells that are specific for SARS-CoV-2 and exactly what they're specific for in SARS-CoV-2. So we're able to assess a significant part of the T cell response in a way that no longer requires live cells, and that changes your ability to standardize and scale T cell measurements.
Would you briefly describe any current partnerships that you're involved in? And if so, how are the trials going, if trials are being conducted?
Sure. So we have partnerships with a variety across many different of the large pharma companies who are making the vaccines on market, but also we're intimately connected with a company called Nycode that's making a next generation T cell vaccine. Along the other company that's doing that is Gritstone, and there we're making I think significant progress on what would be you would call a universal booster that will be invariant no matter what the next strain of SARS-CoV-2 is, and should really create a broad immunity that could last for a considerable period of time on the T cell side, so that even if a new strain comes around that spreads wildly, our immune system should keep us from getting severely sick, and we can move to an endemic state where it's maybe causes cold-like symptoms, but we'll live with that, and it won't cause the kind of the hospitalization and death that the SARS-CoV-2 has with people without a broad T cell immunity. So I think we're really excited about this partnership, and we think that I think the world might go in that direction. We've heard rumblings from some of the really big vaccine makers as well that they're thinking about going in this direction as well. So we definitely think T cells are having their day, which is important for us getting back to a regular life.
Give us a website where we can learn more about adaptive biotechnologies, if you would.
So the easiest way to get there is just adaptivebiotech.com. There's links to everything, that all of our different directions in SARS-CoV-2 as well out there.
Right. Well, Dr. Robbins, I appreciate your time this morning. Thank you so much for joining us here on Health Professional Radio.
Oh, my pleasure. Thank you for having me.
You've been listening to Health Professional Radio. I'm your host, Neil Howard, in conversation with Dr. Harlan Robbins of Adaptive Biotechnologies. Audio copies of this program are available at hpr.fm and healthprofessionalradio.com.au. You can also subscribe to the podcast on iTunes, listen in, download at SoundCloud, and be sure to subscribe to our YouTube channel at youtube.com/Health Professional Radio.